lots of different signals. But importantly in plants, both of these decisions are irreversible. So in the decision of a plant, for example, you can't ungerminate. When you flower, you can't unflower. So when plants make decisions, they're one-way decisions. So when we make decisions, for example, if we decide to get married, we can decide to get divorced. Animals have different types of decisions that they can make versus plants, and we can reverse ours often, and plants cannot. And that's important, and I'll explain to you why in a moment. So going back to what I said at the beginning, genetics versus cells versus tissues. In genetics, we have molecular interaction networks, which can explain what occurs within individual cells, but plants themselves are not single cells. So the question we're asking here is, how do collections of cells process information? So to do this, we're asking this question: how do plant organs process information from the environment? We're taking a view to computers to see whether or not the way computers have been designed can teach us about how it is that plants are working. So towards this, we're taking a computational perspective of plant development. So we're formalizing the system. And the way we're doing this is to use the language of computers and explaining that in the context of plant development. So we have inputs and we have outputs. So in the case of the inputs, these are the different signals from the environment. And then the outputs are the transitions, like phase transitions, for example, developmental phase transitions such as seed germination or flowering, but also cell fate. The software that is running on implants is what we call a program. We already call them genetic programs, which is effectively what we call a software anyways, which is a computer program, but we have genetic programs that are running. So that's the software. And then finally, the hardware. This is the cellular context in which the programs are executed. So I'm talking now about the organs themselves, the cells, which have various compartments within them, and the tissues, which are collections of cells which are coming together to form organs. So we have inputs, outputs, software, and hardware. So now distributed computation. Distributed computing is a distinct class of computing which has arisen, I guess, quite a long time ago now, but it's really gaining in popularity. So in the original computers, they had something called a CPU, a central processing unit, and it was the architecture was such that there was a single chip and all operations were performed on one single chip. And then for those of you old enough to remember, somewhere in the late 90s, early 2000s, all of a sudden they started selling computers which had something called a duo core, and it would say duo on the computer. And instead of having one chip, there were two chips. And that was the dawn of mass distributed computing. So the idea now is, instead of having one processor perform all the operations, you start distributing the operations across multiple chips. So here this is illustrated on the left. You have lots of different processors, and each individual processor is performing an operation and generating an output and relaying the output to adjacent processors. So they're performing operations, reaching conclusions to their computations, and exchanging their outputs. And then what happens is they collectively come together. All of the outputs of all of these operations come together and generate what's called the consensus, which is like a unified final computation. So we can think of a multicellular tissue in the same way. So within each individual cell, you're running a computer program, a genetic program, and the genetic program is performing an operation, it's receiving inputs, and it's executing a program, and it's generating an output. And it's in the case of the outputs, these are mobile agents, and we know of many of them in plants. So in plants, the mobile agents of computation include ions, peptides, mRNAs move from cell to cell, microRNAs, proteins, hormones, etc. And they're moving either through plasmodesmata, through transporters, or through the apoplast. Now, examples of distributed computing in computing architecture and hardware, processing processors, central processors, are distributed now. There's multiple chips on machines nowadays, but if you're into gaming, the NVIDIA gaming cards, the GPUs, are distributed the things that you're using. I mean, blockchain for the ledger that's kept for cryptocurrency is distributed. The internet itself is distributed, all the various servers which are linked up to one another. And there's a whole bunch of reasons for being distributed as being advantageous, including robustness and as well increasing computing power. But we're here to talk about plants and not about cryptocurrency. So I will proceed with this presentation. Okay, so is information processed in plants in a distributed fashion? Well, we had an interesting example of this coming from stomata. So I'm speaking to a plant science audience today. I will not explain what stomata are. Well, briefly, they're involved in gas exchange in plants. But there's a trade-off in stomata behavior. So when they're open, you can acquire CO2, but you're also losing water. So CO2 in is good, losing water bad. When they're closed, you cannot acquire CO2, bad, but you conserve water, good. So there's a need to optimize this trade-off between CO2 acquisition and water loss. Using thermal imaging, Keith's MATLAB looked at the surface of the leaf, and you can see this red patch here where my cursor is. This is a time lapse, so you see it's starting here, and then over time the size of this patch is growing in size. So what this is showing us, this is showing the temperature on the surface of the leaf, and this red color is a proxy for when the stomata are opening. So we can see here the stomata are opening not randomly, individually across the surface of the leaf, but they're actually opening in a coordinated fashion. So there's a propagation, there's a wave of stomata opening. So what this means is there's coordination between the dynamics of stomata across the surface of the leaf. So what this means is there's a collective behavior in the stomata. The stomata are not acting autonomously, but they're communicating with one another. And this is being done through a distributed computation, and there's even the algorithm has been described called the GKL, cellular automaton. And it's an algorithm describing the relationships between, remember how I described the processors are connected to one another and communicating? Well, here the stomata are communicating to one another and leading to a propagating behavior. And this is acting to optimize this CO2 water loss balance across the surface of the leaf. So this was a really interesting first example of how distributed computation can be occurring across the surface of a leaf. So something I've worked on for years is seed dormancy, seed germination. And here's a schematic showing the hierarchy of events that controls the germination of seeds. So we have signals from the environment, which include light, temperature, nutrients, even smoke. This is a dormant seed here. So these are perceived by the dormant seed, and each of them have their own receptors, which initiate downstream signaling events and signal integration. And then finally, this converges upon a switch, which is in a no or yes position. So initially, when the seed is dormant, it's in the off position, it's stopped. And eventually, a certain threshold is passed within the integration of all these signals, where this stop signal is then flipped over to a go signal and germination is initiated. So the computation that's occurring here is we have multiple inputs from the environment, which are being integrated to generate a single output, which is the decision to terminate seed dormancy. So this is mediated, I'm going to guess that many of you are familiar with this, by a hormone thresholding mechanism through the mutual inhibition between ABA and GA. So this creates a discontinuous switch. So you have ABA inhibiting germination and GA promoting germination, and the one is inhibiting the other. So what happens is you're either dominated by the one state or dominated by the other state. So when ABA is stronger than GA, it's blocking the GA, but when ABA becomes weak and GA grows, the GA becomes dominant and blocks ABA. So you're either in one state or the other state. So we've known this for 40 years, but what we didn't know before we started looking at this, which is where these events take place within a dormant embryo. So which cells within the embryo are mediating this ABA-GA antagonism? So to answer this, we generated reporters for the genetic constructs of genetic components of each the ABA and GA signaling components, and we localized them within the dormant embryo. And to summarize a tremendous amount of work in one picture, this is it. Here is an Arabidopsis embryo. These are the cotyledons, the embryonic leaves, and this is the embryonic axis. And this here is the embryonic root. And whoop. And false colored in here in Photoshop, this is not real data, are the locations of the cells which respond to each ABA and GA. So in ABA, the cells here are colored red, and in GA, they're colored green. So the first thing that really struck us is that there's actually a very small, of the roughly 14,000 cells that are in the Arabidopsis embryo, it's actually a very small number of cells that are responding to the hormones ABA and GA. And also, so what this represents here, this small collection of cells is effectively a decision-making center, whereby you have not the whole organ making these mutual inhibition, which is mediating decision-making, but there's actually a very small subset of cells. And the other thing that's striking is the fact that the cells responding to ABA and GA are not the same cells. So you have these cells on the root cap and epidermis responding to ABA, and the cells in the vasculature responding to GA. So what this is telling us is that ABA and GA responses are spatially separated in mutually antagonizing signaling centers. So what this means is that you have cells which are inhibiting germination and separate cells which are promoting germination. So this model of the ABA-GA antagonism at the molecular scale is actually incomplete in the sense that that antagonism is not occurring within the same cell type. Cool. So then we started looking around and saying, okay, well, if you have a signaling system, a decision-making system, where you have certain cells promoting a decision and other cells inhibiting a decision, are there other examples of that in nature? And it turns out that there is, and the place where that exists is actually within the human brain. So the human brain is also distributed in its architecture. We have different regions of the brain involved in different processes. And I'm not a brain expert, so I'm not going to try and pretend like I can explain that all to you. But I can describe to you one aspect of it, which I've read about, which is relating to human motor movement. And this is involving three regions of the brain called the basal ganglia, cerebellum, cortex, and it's a loop. So here we have a cross-section of our brains. And here we can see that the cerebral cortex is up on the top here. This is where we do all the smart stuff. And then there's the basal ganglia here, which is a little more basic, but it's down here. And then there's the cerebellum, which is even lower down in the brain. But importantly, these are three anatomically distinct parts of the brain. So if we look at it schematically here, we have the basal ganglia, which is actually negatively acting to stop human motor movement relating to, the physical movement of the body. So the basal ganglia is inhibiting motor movement. The cerebellum is acting to promote it. And these are each being integrated within the cerebral cortex. So what this represents is a group of cells in the basal ganglia acting negatively and a separate group of cells in the cerebellum acting positively. So this is the analogous anatomical feature between this motor movement loop and the ABA and the GA cells. And it's been proposed that this physical separation between the basal ganglia and the cerebellum, what it does is it introduces a time delay because there's a physical separation between the cells and that's not all occurring within one cell. It takes time for the signals from the negative and the positively acting centers to reach and be integrated within the cerebral cortex. So this spatial separation introduces a time delay, and it's been proposed that this filters noisy inputs. So because we exist in a noisy environment, in nature is a noisy place, we're constantly receiving many different stimuli. If we reacted instantly to every positive stimulus that we received, we would be compromised in our capacity to make accurate decisions, because we would just constantly be getting triggered in order into making a decision. So there's something known as Fitts' law, which was proposed in 1954, which is this notion of a trade-off between speed and accuracy. So you don't want to be too fast or too slow, but at the same time, you don't want to be inaccurate. So we can take major life decisions, going back to marriage. Marriage is a great option for illustrating these concepts and the speed versus accuracy trade-off. So you can be with a person and you could decide, oh, this is a good person for me to marry. But then perhaps if you do this too quickly, you may have not found the correct person, or if you do this too late, maybe you cannot find a person. But in the sense that there must be there's a trade-off between the choice that you make in terms of how quickly you make it versus how accurately you can make it. And there should be some sort of an optimum. And where that optimum lies depends upon the context that you're existing. So in the case of humans, like I said, we are able to reverse our decisions. So the accuracy is not as important as it is in the case of plants where decisions cannot be reversed. So this is something to keep in mind. So going back to the noise filtering, we then asked the question, is the spatial separation in seeds acting in the same way as it is in the human motor decision-making loop? So here we're asking the question whether noisy inputs are being filtered by the spatial separation present between the ABA and GA responding cells. So what we did is to ask whether seeds are also filtering noisy inputs, we took dormant Arabidopsis seeds and applied low temperatures, which are a dormancy breaking treatment, and then provided either continuous cooled or interrupted cooled. So the idea would be that if seeds were filtering noisy inputs, if we provided continuous cooled, that would break dormancy more effectively than providing interrupted cooled. Because if the interrupted cooled is being filtered, it won't be as informative, as constructive towards the breaking of dormancy than giving it a continuous signal. A continuous strong signal will be better than an interrupted signal. So here are the results. On the right-hand side in the dark blue, we have the seeds, which are dormant, and they're germinating about 10%, fine. And then we provided either 96 continuous hours of cold, which is in the light blue, which gave us about 60% germination. So that's 4 straight days of cold, broke dormancy to about 60%. We then provided cold, either with 24 hours of cold, followed by 24 hours of warm, and 24 hours of cold and warm for different numbers of intervals. So here are the numbers of intervals here, which is numbers of pulses. So here, this is just one 24-hour cold interval. This is, and then two 24-hour cold intervals. And then by the time we gave the seeds 3 24-hour cold intervals, they were germinating up to 95%. And if we gave the seeds 96 hours of cold, instead of them being continuous in 4 one-day installments interrupted by warm, we got 100% germination. So what this meant is that our hypothesis was wrong, that seeds in fact are not filtering noisy inputs, but they're actually using noisy inputs. So the seeds are preferring interrupted cold rather than filtering it. So what this is indicating to us here is that this variability is acting as an instructive signal in the context of seed dormancy. So in development, noise is something that needs to be filtered in order to create robust outputs. So say, for example, when a plant is developing in a noisy environment, you want the plant to be as robust as possible in terms of the morphological and physiological characteristics that it has, despite the fact that it exists in a noisy space. You want it to be a robust entity. However, in seeds, a variable environmental input is actually instructive and not a destructive signal. So the seeds are looking for variable inputs, which is something that was rather unexpected. But however, this isn't a completely illogical or unreasonable evolutionary adaptation, because in the places where Arabidopsis exist, for example, in the UK, we see larger daily temperature fluctuations in the spring and in the autumn than we do in the summer and in the winter. So in the life cycle of Arabidopsis, the seeds are germinating in the spring and in the autumn. And at this time of year, for example, we see greater daily highs and lows than we do in terms of the temperature than we do in the spring, in the summer and the winter. So what this is an indication for the seeds is what time of the year it is in terms of the transitions between the seasons, as opposed to the seeds using just an absolute amount of cold over time. So it's quite a sophisticated mechanism that they've evolved, and it can tell them what point of the year they're existing in. And data I won't show you is that the fact that this complicated waveform transformation of temperature requires a spatial separation of the signaling centers. And if you want to read more about this, you can read about it in this paper here. And I'll just skip over this result. Okay, so that is distributed computing in plants. Now I would like to shift topic and start talking about the hardware, plant hardware. So the context that genetic programs are running within plants. So plant organs are collections of cells that are glued together by shared cell walls. So unlike in animals, the cells in plant organs don't migrate, they don't move around. So we can look at them as integrated systems of interacting cells, or spatially constrained three-dimensional cellular networks. So by spatially constrained, I mean they're unable to rearrange themselves. So cities are another example of a spatially constrained network of roads. So cities are networks of roads, but the roads themselves are unable to move. So when a road is in a place, it can't move. Now cities, largely speaking, are two-dimensional because they're planar, because they exist on the surface of the earth. Okay, maybe you have some overpasses or you have a subway. But in the case of plants, we can think of them as being three-dimensional networks because you have cells existing within 3D space. And these are the templates upon which information is being processed. And by that, I mean, it's in these three-dimensional networks of cells that all these genetic programs are running and exchanging information with each other. So we developed a method in the lab to map the connectivity of cells in plant organs. I'll take you through this really quickly, which is that we take the tissue and we stain the tissue with a fluorescent cell wall stain and then segment the cells in 3D and then extract the connectivity between the cells using 3D image analysis. So what we do is take the tissue and then abstract the tissue into a network where the nodes are the cells and the edges are the physical associations between the cells. So the network is representing how the cells are physically connected to one another. And once we abstract the tissue into these networks, we can then use network science to characterize how the cells are connected to one another quantitatively. So we're able to discretize cellular organization, and we can quantify patterns in tissues using network science. And what's important about this is that we have the whole organ represented, fully represented. So these are high confidence data sets. So in other types of network types, for example, if we're looking at like protein-protein interaction networks, the data is often incomplete, but also subject to artifacts and inaccuracies. These are actually extremely accurate. So we can look at the entire system all at once and have a very accurate overview of it. So just to give you an idea visually of what these networks look like, this is an Arabidopsis hypocotyl. This is laid out in 3D. And the balls are the cells, and the green lines are the physical connections between the cells. And the different cell types have been identified, and they've been given different colors. Oh no, where's the video? Here we go. And they've been given different colors. So I'm just going to spin this around. So this is just the layout of the network to give you an idea of what the data look like behind what I'm going to present to you. And this is a hypocotyl, and it's spinning around, and it's just to show you, isn't this pretty? But also to give you an idea that the network is just describing how the cells are stuck together to one another. Okay. So in terms of abstraction, here, this is part of the image analysis. So this is the cells of the hypocaudal, and on the right is the network. We go from these gigantic image stacks, which are like about 2 gigabytes, and just build them down, abstract them down into text files, which can be as small as 160 kilobases, which is over 10,000-fold compression of data, going from a gigantic image file down to a text file describing a network. So this is a lot of abstraction. So I'd like to say, oh, this was my original idea, but it wasn't. It can be traced back to this man here, whose idea to map cellular connectivity came about over a century ago. This is one of Spain's most famous scientists. His name is Ramon y Cajal. And here he is with his microscope. And he's looking a little bit sad. And the possibility for his sadness is that he really wanted to be an artist, but his parents wouldn't let him be an artist because they were worried that there wouldn't be any job for him as an artist. So he became a scientist, and then along came this microscope. But he had this brilliant capacity to combine his love of art and science and microscopy to create what are now known as Cajal diagrams. And what he did was take the nervous tissue of animals and stain it with a Golgi stain, a silver stain. And then he would look in the microscope and draw what he saw. And importantly, what he saw was the fact that the neurons within animal tissues were actually connected to one another. So we take this for granted now. But if we think back at the end of the 19th century, they didn't know this. So this was pretty remarkable. So what he drew were these diagrams describing how the neurons were connected to one another. And he drew these diagrams now known as Cajal diagrams. And the real insight from this came from the fact that the cells were existing as systems of interacting cells that came together to create complicated systems greater than the sum of their parts. And he even had the insight to realize, if you look at this picture down on the bottom right, that the neurons had a directionality to them. So you can see he's drawn arrows on these neurons to show, because as we know, the neurons do actually fire in specific orientations. So he was able to derive this just from the anatomy of the neurons. And he ended up winning a Nobel Prize. He also had an organelle named after him, which he identified, known as Cajal bodies. So a fantastic scientist, but the notion of mapping cellular connectivity can be traced back a long time now. Sidney Brenner's lab continued on with this work in 1985, mapping out the neurons of the worm C. elegans, and created a network describing the connectivity of all of these neurons. This is back in the 80s. And this was amazing because once they had the complete network of all of these neurons, they were able to predict the function of individual neurons just based on where the neuron was positioned within the global topology of the nervous system. So they were able to predict neurons involved, the function of individual neurons involved in smell, involved in moving backwards, involved in wiggling. So this is pretty cool. So the idea here is that if you map out the entire system, that you can then use network science to predict the functions of cells within complicated multicellular systems. So then we asked whether or not we could do the same thing in plants. So now that I mentioned that we can map global cellular interactions, this is the equivalent of mapping the cellular circuitry of the plant, the multicellular circuitry. And then we can ask the question whether we can predict the movement of information and the functions of cells based on their positions within multicellular tissues. So we can perform, say, a couple different basic analyses using network science. The most simple one is the degree. The degree describes just the number of neighbors that a node has within a network. So to use Facebook analogies, your degree in the social network known as Facebook is how many friends you have on Facebook. So this is a very simple measure. It tells you how many people you are directly connected to. And in the case of a plant cell, it'll tell you how many cells a node is directly connected to. So while that's kind of informative, it can tell you how popular you are. It's a very local measure. So just because you have a lot of friends on Facebook doesn't mean you're an influential person, because you could be connected to many people, but if your influence is, if their influence is in turn also only very local, then your influence within the social network is actually very limited. So we can look at other measures of connectivity in networks, and I'll just describe them at a very high level. This notion of betweenness centrality and random walk centrality. And what this describes is nodes within networks that lie upon shortest paths between other nodes. So if you want to go from one place to another, you will follow the quickest way or the shortest path. And it's the nodes that you go along the way that you pass through that represent the optimized routes for going through the network. So those optimized routes actually have a lot of influence over the broader network, because if they are the best way to move, or the most connected ways to go between one node and another node, then they're now controlling the movement of information across the network. So if you think of a city, the most busy roads, I'm sorry, I can't think of any road names in KL, but I can imagine that there are certain highways that everybody takes. And if that highway is closed, then it causes a really big problem, because there's so much flux through that one road that if it's closed, there's just almost not enough roads. Nobody can move. So that one road is actually a super important road. So if you think about that in a social network context, if you're somebody who knows many other people or knows important people, then you actually can carry, without yourself being in like the important person, but if you're connected to the important people, then you can control, you can exert a lot of influence over a social system. The same is true in a plant or in any other network. So these are nodes that lie upon shortest paths between other nodes. If you want to read more about that, you can read about it in this paper here. Okay, so we can map the connectivity of cells and tissues. We can get networks, we can analyze the networks, and this has been done in C. elegans, for example. So now we're going to ask the question, can we do this in plants? Can we map the connectivity of cells in plants and predict the functions of cells based on how they're connected within tissues? So can we uncover structure-function relationships in plant organs using this connectionist approach? So we did this using the Arabidopsis hypocotyl as our test case. So in the case of the hypocotyl and then the root, there are two different epidermal cell types that are present. You have the trichoblast epidermal cell type, and the atrichoblast. So the trichoblast cells are cells that make root hairs and they take up nutrients. The atrichoblast cells do not make root hairs. So trichoblasts make root hairs, and here you can see them indicated in green. This is a cross-section of a root, and you can see a hair coming out, and it's an H, and atrichoblasts are in blue, and they don't make hairs. So key differences between them is that these trichoblast cells are lying on top of two underlying cortical cells, whereas the atrichoblasts are just sitting on top of a single cortical cell, one in cross-section. So we've known about the existence of these cells for many years, and we know a lot about the genetic networks that lead to their creation. But what has been less well understood is, why are there two cell types, and why are they arranged in the way that they are? So we know that this is always the case in every root that we do a section. They have these identities, they have this organization, but the fact that they are that way, it's less clear why they're that way. So we decided to address this using this network-based approach. So we took Arabidopsis hypocotyls and we identified the cell types, and we separated the trichoblast and atrichoblast cells, and then looked at their topological properties. So the first property we looked at was this local property of degree, of how many neighbors they have. And we can look here in Colombia at the trichoblast and atrichoblast, and the number of neighbors they have is plotted as a frequency histogram here. And the atrichoblasts are shown in light green and the trichoblasts are shown in dark green. And what this is showing is that the trichoblasts, the cells that make hairs, have more neighbors than the atrichoblasts, the cells which don't make hairs. So what this means is, this is actually not surprising because, as I mentioned, the trichoblasts are sitting on top of 2 underlying cortical cells, whereas the atrichoblasts are sitting on top of 1 underlying cortical cell. So it's not unpredictable that the one sitting on top of 2 cells has more neighbors than the one that sits on top of 1 cell. But then when we look at between and centrality, which is looking at the number of shortest paths that the cells are lying on, the atrichoblasts, which have fewer neighbors, are actually lying upon more shorter paths than the trichoblasts. So what this means is, despite having fewer neighbors, the atrichoblasts are actually faster ways of going from point A to point B than the cells that have more neighbors. So this is a bit of a counterintuitive result. So this represents previously undescribed conduits of reduced path length within the non-hair cells. So the reason they're conduits of reduced path length is because they lie upon more shorter paths than the non-hair cells. So what's the point of that? That's kind of cool, but what's the functional relevance? Well, to look at that, we performed molecular transport assays. The way to do this was we took seedlings, 7-day-old seedlings, and transferred them onto media, which contained the small fluorescent molecule called fluorescein, and then imaged the hypocotyls using confocal microscopy. So the idea was we put the seedlings onto media which contained a fluorescent molecule, and then looked to see where the fluorescent molecule was moving as it went up the plant. And this was the result, and the result was dramatic. So here in white are the cell walls, and in red is the fluorescein molecule. And what we can see is in the A cells, the non-hair producing cells, we see a lot more fluorescent molecule than we do in the T cells, which are making root hairs. So this is a bit unusual because the trichoblasts are the cells which are making root hairs and presumably involved in the uptake of the fluorescent molecule from the media. However, despite the fact that these cells are involved in taking up the fluorescent molecule, there is more fluorescent molecule in the adjacent cells. But this is in fact what we predicted from the path length analysis. Because these cells are lying upon more shortest paths, these represent a more rapid conduit for the movement of this fluorescent molecule than these. So despite the fact that these cells are involved in uptake, these cells are actually better at transport. So what we have identified here is a topological proxy, which is betweenness centrality, which predicts molecular movement through complex organs at single cell resolution. So we can take a network of cells and do this network analysis and predict exactly where the molecules are going to move through the tissue. It represents an optimization in epidermal cell patterning for longitudinal molecular movement, and the optimization is represented by the fact that they're lying upon more shortest paths. And finally, to talk about this bridging of structure and function, is it also represents higher order properties of multicellular patterning. So the structure and function, I asked the question, why are there 2 cell types? Well, it's a division of labor. So if you have the hair cells, which are involved in the uptake of solutes, they're busy taking up the molecules from the soil. That's their function. But then they have next to them their neighboring cells, which are the non-hair cells, but then they receive these molecules from the hair cells, and then their patterning has optimized them for longitudinal transport of solutes. So there's a division of labor of uptake and transport between the hair and the non-hair cells. Great. So if you'd like to read more about that, you can read about it in this paper here. And I'm just looking at the time, and it's 8:42. Would this be a good place to stop, do you think?
You have more? You can continue a bit if you want.
Oh, I could talk all day. I have so many slides.
You can go for another 3 minutes or so, I think.
Let me have a look. You know, to be honest, it gets pretty complicated. But fundamentally, what I wanted to say about this is the communication between cells is controlled by often a very common method is through plasmodesmata. The symplasm, the cytoplasm, and endoplasmic reticulum between adjacent cells is being controlled by the opening and closing of these plasmodesmata. And we believe that this represents a very important way in which plants are controlling their overall processing of information from the environment. There's an interesting link to technology, which is this notion of I'm struggling with my mouse. It's very sensitive. If I just touch it, even a little tiny bit, it flips the slides. Sorry for making the slides jump all over the place, everybody. Right, I'm gonna just keep my hands here and not flip the slides. Right, this technology called a field programmable gate array. So they use these types of distributed chips when they make satellites. And the reason they do this is they're able to, it's a distributed architecture. So you have lots of different processors on a circuit board, but the way that they are connected to each other is it's not hardwired. The ability, you're able to dynamically reconfigure the circuit. So the way the chips are stuck to each other can be changed. And the reason they do that is when they send a probe into space, the missions that go for decades, the probes that are out very, very far away collecting images in faraway places were launched, I don't know, 30 years ago, some of them. So if you think of the technology and the software that had been created 30 years ago, we think of, I mean, cell phones didn't exist, for example. The engineers in NASA had the foresight, because they're very clever people, to put together these FPGA architectures that say, we're not going to give it just one chip and say, this is the only way you're able to process information, but they gave them chips that could be reconfigured. And what that does is increase the computational complexity. So the ability to rearrange the architecture to create different distributed circuits increases the complexity. So a great example of this is one of these space telescopes that got released a long time ago. And they didn't anticipate that the space telescope would go for so long, and it keeps going and going, and it creates a lot of images, and it's creating so much data that it was impossible to send all of the data back to Earth because the transfer time took too long. But since they released it, they were able to create a new compression algorithm to make the images smaller. So they were able to through the radio to send the computer program for the compression algorithm to the probe in space, and then reconfigure the hardware, the chips, to optimize the hardware to run the new software. And then they're able to send more images back to Earth, and we can see more into deep space. So we think that plants might be doing something like this, and dynamically reconfiguring how the cells are communicating to each other in order to increase their adaptability to the environment. Okay, I'm going to stop here. Why does this matter? Well, the context that genetic programs operate within impacts their output. So what we're trying to do here is to understand how the hardware, how the configuration of cells is controlling the outputs of genetic programs. So we have really limited GMO success stories, and I think a reason for that, I would say no GMO success stories, except for putting bacterial genes in plants like Roundup Ready or BT. And I think the reason for that is that we have levels of complexity in plants that we don't fully understand. So if we just only try to alter the genetics in the molecules, that's not enough information in order to be able to predictably and meaningfully reprogram crop plants. And this is representing a novel approach to re-engineer plant development. So I'd like to thank the people that did it. This is work that was done in Birmingham. I'm now in Warwick, but I'll thank the Birmingham people, Matt Jackson, Salva Duran Nebreda, Hao Shu, Petrostam, Alec Topo, and the people who have given me money, the BBSRC, the LibreHume, the Human Frontiers Science Program, Warwick, and I'm a fellow at the Alan Turing Institute, where I'm working with artificial intelligence scientists to see if we can take algorithms from plants and learn from them to use them in computers. And thank you very much.
Okay, thank you, George, for the fascinating talk. So now is the question and answering session. I'll read out some of them from the chat box. I think there's one from posted by Dr. Imelda. Not sure it's from Facebook or from her. The question is, can ABA and GA regulate the carbon dioxide and oxygen in the plant? I think
certainly,
that's your feed. Yeah.
Certainly ABA plays a major role in controlling stomata opening and closing. I think if GA plays a role, it would be minor. The control of stomata from a hormonal level is a different system than the antagonism between ABA and GA in seeds.
Okay, the next question is from Dr. Nisha. There are so much of genetic variables that affect stomatal opening. She would like to know whether the parameter that fit into the genetic algorithm established for the stomata opening, what are the parameters that fit into the genetic algorithm for the stomata opening?
So I think we know a lot about that if we were to look at only one pair of guard cells, one stoma. There's a lot known about how signals are used to control the opening and closing of a single stoma. Single stomata, I should say, whatever. Stomata?
Yeah, stoma.
Stoma. Thank you. So then how it's done on the collective level, I think, is less well understood. Like how is it coordinated across the surface of the leaf? I don't know. Sorry?
Okay, the second part of the question is that, is the genetic algorithm specific to the plant species or the leaf type or the leaf developmental age?
Sorry, I'm not sure I understand the question.
The first part of the question is about the genetic algorithm, right? About the stomata opening. The second part is whether this genetic algorithm is specific to the species or the leaf. If I understand correctly, the question is like whether the genetic is conserved or is specific to the context.
I'm sorry, I don't know. Question.
Okay. So, okay, there's no more question in the chat box, but I have my questions. It's fascinating about the different signaling center in the seed, the INBIOSIS embryo regulation during the seed dormancy. So, how the signal is propagated from the roots? Is it through the auxin signaling, auxin transport? So you show that the centers are based in the roots, right? But it's controlling the whole embryo.
What's controlling the decision to initiate?
How the signal?
Yeah, this is the bit that I skipped over. It's controlling the decision to initiate growth. So in the dormant state, the cells are just sitting there dormant, and they can sit there, I don't know, in the case of Arabidopsis, 3-5 years. Some seeds can sit there for 10,000 years. There's lotus seeds that are, you know, in Buddhist temples that are still sitting in jars that have been there for thousands of years. But the way the signals, the hormones are transported, it's actually not through auxin transporters, but there's actually several transporters for ABA and GA, and some that even transport both ABA and GA. There's one called NPF3 that we looked at the study, but it's not through PIN proteins or OX influx carriers. They're different classes of transporters.
Okay. So any more questions from the audience? Any question from Facebook? I'm not sure. Okay. So I think it's fascinating you try to find analogy between the computers and also the plant organ development. So what else you're working on at the moment apart from the hypocotyls and then the seeds? Are you still working on the seed modeling?
A little bit of seeds. We're interested in how the plants are processing temperature. So we saw this really interesting, what I presented, that the alternating temperatures are more interesting for the plant than a constant temperature. This was really a big surprise. You think of a plant as just, you know, maybe not so complicated, not so clever. I still don't think they're clever. I don't think plants are clever, but they can do this one thing, which is pretty cool. But then we're interested as well in how the plants are using temperature to flower, how to break like invernalization. That's of interest. And we also have another project looking at bud dormancy. So when you maybe don't have this in Malaysia, but in this part of the world where it gets cold in the winter, the buds have dormancy, and then in the spring they break dormancy and make leaves and make flowers. So again, how the temperature is being processed within the cells and the dormant buds.
I think in tropical country we have a different phenomenon. We have a very hot season and then the very like rainy seasons, and this kind of seasonal effect actually have this influence on the flowering of the tropical species. So instead of temperature, there's other factors. I think those are still like understudied. There are many fascinating tropical species regulation that is not so common among the model temperate plant species that's reported today.
Yes, that is really interesting. So is it not the case that the plants will drop all of the leaves at the same time?
Usually the evergreen, like the tropical species are evergreen, so they wouldn't like have the abscission of the leaf like the temperate species. So it's evergreen. However, there is regulations as well. Like for example, we can see that the flower blooming simultaneously for a certain location for the genetic regulation of it is not clear in a way, because usually it's like we follow the very hot season and then rainy season, and then there will be these fruiting seasons for the tropical tree, like when we have the durian seasons. Then we have a tropical the flower seasons. So we have a certain kind of seasons that is not regulated by temperature or the day length as in temperate species.
So it's more by the water.
So we have another question.
Oh, sorry, yeah.
I'm not sure. that part I'm not sure. There's something to study about. There's a question from Dr. Ng Chan Leong regarding the phenomenon of stomata open close in a regional manner. Since plants does not have neuron system, what kind of signal transduction system that may plant decide which region to open and close?
Yeah, that's a great question. So this isn't my work, but the people who did the work, they were looking at, the question is what is the signal between the stomata? So it's been proposed that it's actually an electrical signal.
Calcium?
So, calcium, yes, could be calcium. But as we know, the plasmodesmata, though, are not connected to the neighboring cells by plasmodesmata. They're symplastically isolated. But it's believed that on the surface of the stomata are these ion channels that transport potassium, I think it is, and these slow anion channels. But it's proposed that the membrane potentials and the signals, which are electric, are coordinating these behaviors.
Okay. So if there are no more questions, we'll close the webinar. So I would like to thank Josh again for the talk. And I would like to thank all participants. Oh, sorry. There's a response from Dr. Ng about if through the calcium or ion, what kind of speed that we are normally expecting? Very challenging questions for Josh.
What's the question? Yes. What's the question?
Continue from the signal transduction of the stomata propagations. So how the if it's through the calcium or iron, he's asking the speed of the signaling.
The speed is, I mean, it's slow. It's like at least 10 to 50 times slower than nervous transmission. Like it's nothing like in animals. This is also not my work, but there's a growing body of literature showing the movement, the propagation of like calcium waves through tissues, but also you can measure it using electrodes, these bioelectric signals. The speed, the time scales, wow, I don't really know, but I do know that it's at least an order of magnitude slower than it is in nervous transmission, if not 2 orders of magnitude, like 100 times slower. Sorry, I don't know the details of the time scale.
Okay, so before we finish off, I would like to invite all the participants to on your video camera so that we can take a group photos. So
cool.
is it possible everyone can on your camera? It's a photography session now. Okay. Any more? Please on your camera. The attendance will be, the link will be posted after this. Don't worry. So please on your camera. Okay, I think that's all. So Dr. Imelda, can you help take the photo?
You can also go.
I'm not familiar. I will leave it to the expert.
Okay, let's smile everyone for the group photo. 1, 2, 3.
So we have 91 participants on the Zoom meetings. And I think there are around 20 or something on the Facebook Live. Thank you for joining us today. Okay.
Thank you everyone for listening today.
Okay. Thank you, George. Hope you have a good day, good Friday, and good Friday to everyone as well. So I think that's all from us. Thank you for the team, the webinar team, for organizing this. Okay, see you all again in the next webinar. Bye, George.